Thanks, Chris, for inviting me to speak in this forum and talk about our paper that was published at the end of 2020. As Liang and Chris mentioned, I'm not new to the Harvard Quad. I was in the Seidman lab many years ago, and I attended similar forums and discussions of labs in the Quad and at Boston Children's. So I'm very fond of that community, and then to be honest, I pretty miss being there, although I know now everything is virtual. So thank you for allowing me to share my research with you. So the title of the talk is Polygenic Background Modifies Penetrance of Monogenic Disease. And I'm going to lead with my conclusions here. By the end of probably about 30 minutes or so, I'll share with you how integrating monogenic and polygenic risk improves genomic risk prediction in coronary artery disease specifically. I think one aspect Chris didn't mention is I'm a cardiologist by training. I'm actually now in the hospital. And so my research is biased towards coronary artery disease genetics, which is what I focus on. But then again, the subject of the paper, we took that concept and extended it into CDC tier 1 genomic conditions. So I'm going to show you how polygenic background modifies the penetrance of monogenic disease in all 3 of the CDC tier 1 genomic conditions. And at the end, we're going to take that concept and look at some of the data that are evolving in that space on how the polygenic background actually contributes to penetrance of many monogenic traits, including some recently published data that came from other groups after our paper came out. So to start, I'm gonna go all the way back to Lebanon, where I started my scientific career, and that's the country where I grew up. And probably there's a lot of pediatricians in the group here who are familiar with a disease called familial hypercholesterolemia. And that disease occurs due to mutations in 3 genes, one of which is the LDL receptor. And then a mutated LDL receptor results in very high LDL cholesterol. This disease was actually first described in Lebanon in familial cases because of founder effects. And that was described 10 years before the receptor was actually cloned. It was back in the 1960s in the university where I studied. So being there, I encountered a lot of patients during my medical school who were similar to this patient on the left side, who are young people coming in with premature heart attacks. This was a 38-year-old guy who was previously healthy and really came in with a big heart attack. And that's kind of was my early introduction to genetics. I went ahead and started looking, why did this gentleman have a heart attack? His LDL was pretty high, 200 milligram per deciliter. Anything above 140 we consider high. And then we extracted his DNA and started looking into his history. It turns out he has multiple family members with early premature disease and high cholesterol. And that was due to a mutation in the LDL receptor. This mutation is a truncating mutation called the Lebanese allele because of a founder effect in Lebanon. And it contributes to more than 50% of the disease in Lebanon. So it was that patient and a few others like him that actually prompted me to study this disease in Lebanon. I ended up starting a registry of the disease. And then as we started doing cascade screening and looking at other members of families, what we started seeing are people similar to this gentleman on the right side who have survived most of their life without actually any coronary disease, despite having the same genetic mutation and despite having high cholesterol at times. So that was interesting, and I think this concept to any geneticist is well known. It's called incomplete penetrance, where people with a genetic mutation do not end up developing the disease, and it's not really fully understood why that could happen. It could be lifestyle factors. It could be the polygenic background has been hypothesized to play a role in this. And when we look at coronary artery disease, it's become clear over the past several years that there are truly 2 models that drive risk. One is the monogenic, which is the traditional approach that I just described, where a single mutation, in this case, it's a defect in the LDL receptor gene, has a large effect in a single biological pathway. And in this case, it's LDL cholesterol metabolism. And that drives cholesterol buildup in the coronary arteries and results in a heart attack. However, past several years clearly showed that there's a different model, the polygenic model, And nice work that came out from my mentors, Sheikh Katharisen and Ahmed Kara, before I joined the lab, was really showing that that this polygenic model actually plays a pretty significant role in coronary artery disease genetics. And this idea is that multiple variants, each with a very small effect, and then acting through many different pathways, many of which we don't understand, but some of which we do, together can contribute to increased risk in a segment of the population. And that segment is estimated about 8% of the population have a similar risk to monogenic disease. So much more common, yet kind of unidentified, untreated segment of the population. And really some of the work from Ahmed Kara and Sheikh Katharisen, again, before I joined the lab, that started in 2018, basically quantified that number into a polygenic score. And it's a number that basically is a genome-wide polygenic score. It's a cumulative effect of all the small variants that contribute to risk. And if you plot that number in the population, you'll have a normal distribution. And when you start looking at the upper tail of the distribution, what you find is that individuals at the high tails of the distribution have several-fold increased risk of coronary artery disease. And since that paper, there's been tons of studies that are coming out showing in different populations and different places around the world that basically this polygenic score really stratifies individuals at risk. And in this example that I'm showing here, it predicts lifetime trajectories of risk of coronary artery disease. This middle one is data from the Malmo Diet and Cancer Center, which had follow-up over a long period of time. And then the reason this is important is that that polygenic score can enable targeted interventions. And there's been so far retrospective data, whether post-hoc analysis of clinical trials or other retrospective data, actually showing that potentially individuals with high genomic risk could be identified, could be targeted in order to reverse disease. So things such as lipid-lowering therapy. Multiple studies have shown that when they look at individuals with high polygenic score, the relative benefit from lipid-lowering therapy is even higher than the relative benefit in people with in the average population. Same thing for adopting a healthy lifestyle. There was a key paper in 2016 New England Journal looking at the intersection of healthy lifestyle and high genomic risk, and showed that individuals with high genomic risk, if they adopt a healthy lifestyle, the risk is actually attenuated, similar to individuals in the general population. So that's kind of in terms of background on polygenic score for coronary artery disease. We simply asked the question, can polygenic background modify the penetrance of a monogenic variant? And this work, again, is published in Nature Communications, is joint work with Wallace Wang from our lab and Julian Homberger from College Genomics, and Ahmed Kara, who's my mentor now, is a senior author on that paper. And when we started with familial hypercholesterolemia as the disease because of our interest in coronary artery disease, but I'll show you how that model also extended into other diseases. And again, this is not necessarily obvious because monogenic risk in coronary artery disease, it's a single causal pathway driven by LDL cholesterol. But the polygenic risk, there's multiple pathways involved, and only about 20% of the loci that are actually involved in the polygenic score are related to cholesterol metabolism. And the remaining 80, most of them are unknown, about 50% actually is unknown, and then about 30% are related to things such as inflammation, cellular proliferation, and vascular tone that are currently not necessarily captured in clinical practice or by a monogenic variant. So in order to kind of approach a study, we did follow two approaches. The first approach is we assembled a case control study from the UK Biobank. And that case control study allowed us to increase the number of cases and subsequently the number of carriers of monogenic mutations. So that was a case control of a 6,000 coronary artery disease cases, 6,000 of healthy controls. And what we did is we sequenced the 3 familial hypercholesterolemia genes, and then we did a clinical curation of all those genes. So the same way if someone has a genetic test right now for familial hypercholesterolemia, you did have that that kind of manual clinical curation by clinically certified lab. And then we also calculated the polygenic score for coronary artery disease. And what we found is that the FH mutation on average, triple the risk of coronary artery disease. And this has been shown in multiple previous studies. It's usually 3 to 4 times increased risk of coronary artery disease for anyone who carries an FH mutation. And then we calculated the polygenic score. And as I said, the polygenic score in the population, if you plot it the same way in our study sample, is a normal distribution. We binned the polygenic score into 3 bins: the low polygenic score, which we defined as the bottom 20th percentile of the distribution, an intermediate polygenic score, which we defined as the middle 60th percentile of the distribution, and a high polygenic score, which we defined as the top 20th percentile of the distribution. And as I mentioned, because the polygenic score, only 20% of the loci are related to cholesterol metabolism, when you look at cholesterol, LDL cholesterol by polygenic score, you don't see any statistically significant difference. So you cannot pick up people with high polygenic score out of a group because of the LDL cholesterol or any other marker. Those people would otherwise be flying under the radar. And then, this is a kind of, one of the key findings here is we looked at how does the monogenic carrier status and of an FH mutation and the polygenic background interplay. And what you're looking at here in blue are the carriers of monogenic FH mutations, and in black are the non-carriers. And again, people with intermediate polygenic score shown here on average about 3-fold increased risk compared to people who do not carry a mutation. But when you start stratifying this by low, intermediate, or high polygenic score, what you found is that people with low polygenic score, despite the fact that they carry a mutation that causes familial hypercholesterolemia, the risk is really attenuated. The odds ratio is 1.3 compared to individuals of intermediate polygenic score who do not carry a mutation. Again, it's almost like the presence of a low polygenic score is protective, and it's attenuating the risk that's imparted by the FH mutation. On the contrary, individuals who have both a high polygenic score and an FH mutation have an augmented risk. And then we took those observations, and then we extended them into a cohort study from the UK Biobank that includes 50,000 middle-aged participants. And the cohort study allows us to look at the interplay of monogenic and polygenic in the context of contemporary care without necessarily selecting for cases. And in this cohort, we had 2,000 individuals who actually developed the disease over a follow-up period of 10 years, and 48,000 individuals remained healthy. When we look at the prevalence of carriers in this group, we got 1 in 250. So again, those are rare monogenic mutations, so you don't expect to see them a lot. 1 in 250 people carried an FH mutation. And again, the same concept here on the left side. For the 1 in 250 that carry an FH mutation, their risk is on average 3 times to 4 times the risk of non-carriers of mutation. And again, the non-carriers in this model would be 249, the remaining 249 of the 250. And really, in the traditional approach, and what clinically is in practice right now is really only looking at the monogenic risk. What we did next is actually we modeled the polygenic score binned into percentiles. Again, here you're looking on the x-axis, 0 to 100th percentile of the polygenic score, which is a common way of showing the polygenic score. What we found actually with this joint modeling is that the curve looks more like this, where there's a substantial gradient of risk depending on the polygenic score, even among carriers of FH mutation. So, zooming in a little bit on that curve, the average risk, and this is here, we're looking at cumulative or probability of coronary artery disease by age 75. The average risk among non-carriers is 13%. The average risk among carriers is 41%. But in reality, that 41% can range between 17 and 78% in carriers, depending really on what their polygenic score is. And the same thing for non-carriers, it ranges between 5 and 32%. So, what we're saying here is that, this is from a conceptual standpoint, we're saying for, incorporating monogenic and polygenic risk to stratify individuals with risk of coronary artery disease, you almost can place someone at, each an individual, if you could only place them on one of those dots and be able to say, well, no, it's not, if you carry a mutation, you're not necessarily 41%. You could be protected if you have a low polygenic score. And that protection could be, could bring you even as low as the risk of the average non-carrier population. I'm gonna wave here. I had the sensor turn off the light, sorry. So, going back to our original question, can the polygenic background modify penetrance of the monogenic variant? I think for coronary artery disease, our answer is yes. And, it made me reflect back on the two patients that I, saw, more than 10 years ago and 15 years ago, I guess, in Lebanon. Could that individual who, who had the same mutation, but had a high polygenic score potentially and be in that plot, and that individual who had the mutation but was really remained healthy, could that individual have a low polygenic score? I don't know for sure, but that was kind of how I'm thinking about it. So it's worth saying though, for coronary artery disease specifically, polygenic background is one modifier of penetrance, going from the presence of a mutation all the way to developing atherosclerosis or blockage in the arteries of the heart that cause a heart attack. It's multiple modifiable and non-modifiable risk factors that are involved. And our group kind of focuses on studying both of those. First, for having an FH mutation, it's going to drive an increase in LDL cholesterol. And there are some aspects that actually modify your LDL cholesterol level that are not related only to the mutation itself. One thing related to the mutation itself and the non-modifier is, do you have one or two mutations? Do you have a mutation in gene A or gene B? What type of variant you have? As we know, the effect size of each one of those is going to be different. In this study, we did not model that because we don't have the power to actually start looking by, by gene and by variant. But I think as data sets grow, we're going to start to have the power of being able to look at penetrance of disease by gene and then by variant. The other thing on modifiable is obviously if someone's LDL is being treated. In this study, we did adjust for treatment of LDL cholesterol. The other thing is obviously the diet and lifestyle and what they eat can affect your LDL cholesterol level. And then regardless of the LDL cholesterol level, going into atherosclerosis, there are multiple pathways involved. Here we showed that the polygenic risk of coronary artery disease, which has multiple orthogonal pathways that are distinct from cholesterol metabolism, can modify that risk. More recently, there's data from Pradeep Natarajan at Mass General Hospital, also looking at somatic mutations that drive clonal hematopoiesis of indeterminate potential. And those kind of driver mutations that occur in adulthood and track with age can also impact the risk of coronary artery disease. And it's one of the aspects that we're looking at. And again, there's a lot of modifiable risk factors. Biggest risk factor for coronary disease is age. And then there's 7 kind of lifestyle behaviors that have can impact risk, whether someone smokes or not, whether, what is their weight, what kind of food they eat, etc. And in terms of next steps, one of the things that I'm working on developing now, and it's subject of a K award that I just submitted, is integrating genomic and non-genomic risk. So bringing that model into the clinic by saying we're gonna take into consideration the monogenic and polygenic somatic risk on the genome side, but also integrated with all the classic and traditional risk factors that we currently use in practice, and build a single model that actually augment our prediction for coronary artery disease. So, as you probably can guess by now, I'm very passionate about coronary artery disease because I see a lot of patients with this disease, and it's something very dear to my heart. But it's important to recognize that this concept of polygenic background modifying the penetrance is not unique to coronary artery disease. And in the paper that we published, we extended it into CDC tier 1 genomic diseases. And the reason we focused on CDC tier 1 genomic diseases is because they are common and they are clinically actionable. So we wanted to focus on diseases that are currently, a lot of adults have them in the US. About 2 million are estimated in the US to have a silent mutation, unknown basically, for one of those genomic diseases. And if identified, there's clear guidelines and recommendations to actually manage them differently and improve their mortality and morbidity. So what are those CDC1 genomic conditions? One is familial hypercholesterolemia due to those 3 mutations in those 3 genes, and we've discussed that. The other 2 are cancer conditions. One is hereditary breast and ovarian cancer. As many of you know, those are pretty famous, BRCA1, BRCA2 genes. Right now, the recommendations are to screen individuals with family history. That is pretty impressive, but no direct public health screening at large at the population level. And then the third one is Lynch syndrome, and that's due to a mutation in one of 4 genes that contribute to DNA mismatch repair. And individuals with Lynch syndrome have premature colorectal cancer at a very young age. So it's very important to recognize if someone has any of those gene mutations, because they essentially, the recommendations for screening would be very different. So going into, what could we do if we identify people at risk of those who are completely asymptomatic and healthy, but living with one of those mutations? With familial hypercholesterolemia, lab testing to check your cholesterol, aggressive lifestyle changes, and lipid-lowering therapy. In hereditary breast and ovarian cancer, frequent mammograms starting at a very young age, and then prophylactic mastectomy, as I'll talk about later on. 50% of women right now who have BRCA1 and 2 mutations elect to undergo a prophylactic mastectomy to prevent the development of cancer. And then Lynch syndrome, very important to have colonoscopy starting at a young age. So for anyone in the population, the recommendation is to start colonoscopy after the age of 50, once every 10 years. If someone has Lynch syndrome, it would be much earlier. It could be even colonoscopies in their 20s or 30s. And again, right now, there's no guideline recommendation for screening the population at large. So it's not a public health approach. It's more of either cascade screening or screening based on clinical criteria and family history, strong family history of disease. So, to make a long story short, we found exactly the same findings in breast cancer and colorectal cancer using our same approach. In the case of breast cancer, we used a case control study from Color Genomics. That's 20,000 individuals, including 2,000 individuals with breast cancer. And then we extended those findings again to the UK Biobank in a cohort of 26,000 women who some of whom were followed, all of them followed over about 10 years, and some of whom developed breast cancer. And really what we found is in the UK Biobank, the average risk of carrying BRCA1, BRCA2 mutation was 35%. That's a risk of developing breast cancer by age 75, compared to the general population of 10%. But again, that risk had a dramatic stratification by polygenic score that ranged between 13 and 76%. I'm going to also comment here that those rates are specific to the UK Biobank, so they don't necessarily reflect general population rates. So individuals in the general population might have actually higher rates. The UK Biobank is known to be biased towards healthier individuals. So what that means is that if this concept needs to be extended to other populations and used clinically, it would have to be calibrated for the rates have to be calibrated for the population of interest. But the concept here is that there's stratification by polygenic score, even at that risk level. And then similarly for Lynch syndrome, for Lynch syndrome, having a mutation brings in a dramatic increase of risks, about tenfold increase in risk, more than tenfold increase in risk here. But again, among carriers, there's a stratification of risk ranging between 11 and 80%, depending on the polygenic score. So, this concept we thought is interesting for two reasons. One, there's a physiologic basis here that we are yet to understand and discover, that we think is going to be important to look at across diseases. Again, as I mentioned, coronary artery disease, 100% of the monogenic is driven by LDL cholesterol, and then only 20% of the polygenic is related to LDL cholesterol. And there are many other pathways involved. And breast cancer and colorectal cancer, similarly, the monogenic causes are thought to act through DNA repair mechanisms, or in colorectal cancer, this mismatch repair. And then when you look at the GWAS, so genome-wide association studies from breast cancer and colorectal cancer, less than 20% actually are involved in DNA repair mechanisms, and then more than 80% act in different mechanisms, or many of which are unknown. And then the second concept is really improving the accuracy of risk estimation. And I think that's a very translational, concept in genomic medicine. If someone has a genetic mutation, it is important to communicate that message that DNA is not their destiny, and then there is more than just a flat average risk, depending on a variety of factors. One of which is the polygenic score, but also lifestyle plays a major risk. I think that one very famous example is Angelina Jolie, the actress, when she went public several years ago with carrying a BRCA mutation and deciding to move ahead with a mastectomy after being coded very high rates of chance of developing cancer. that went public, and actually a lot of people followed. You see a spike. If you look at the data of prophylactic mastectomy, the rates actually followed after the Angelina Jolie phenomenon. It's called the Jolie phenomenon. And I think, the concept here that we're bringing is that that's not an average risk. Not everyone who's going to have a BRCA mutation are going to have exactly the same risk. And that I think is an important concept from accuracy of risk estimation. It's not all ready for clinical practice for the variety of reasons. I think worth highlighting that one of the important reasons why polygenic scores are not 100% ready for clinical practice, and it's obviously it's an area of strong research in the space, is the Eurocentric bias in polygenic scores. And because the data required to build polygenic scores comes from genome-wide association studies. And genome-wide association studies are markedly biased for individuals of European ancestry in a disproportionate way compared to the world population. What results is that actually most of the polygenic scores in multiple diseases actually have a reduced performance in individuals of different ancestries, particularly important in individuals of African ancestry. And there is a concern raised that actually, if you start using polygenic scores in clinical practice, you could potentially worsen healthcare disparities, and that's a very important concern. So there's a lot of effort in that space nowadays. On our side, we looked specifically at in a recent work that we published on the trans-ethnic transferability of the CAD polygenic score. It's a polygenic score still developed in individuals of European ancestry, but we basically just quantified in a meta-analysis how much does it actually apply to individuals of non-European ancestry. So that was a meta-analysis of 30,000 cases and about 500,000 controls across 5 ancestral groups derived from 6 studies. And really what we found is, when we combined all individuals of European ancestry across studies, the odds ratio for standard deviation of the polygenic score, and that's a comparable metric that we used for that association, is 1.6 in individuals of European ancestry. When you look at individuals of South Asian, East Asian, Hispanic ancestry, there's variability, but it's pretty close to individuals of European ancestry. So we think that the polygenic score, at least from this study, appears to be portable to other ancestries. However, it does have a reduced strength in individuals of African ancestry, and the odds ratio was 1.25. It is still strongly associated with coronary artery disease in individuals of African ancestry, but the strength of association is going to be lower. In the last segment of my talk, I'm just going to highlight some other examples that came from not from our group, looking at that same question. Many of which are starting to come up now in studies over the past few months that I think are super exciting. And the question here is this concept generalizable to to other monogenic traits? And I think to a certain extent it could be. It really depends on the trait, depends on the strength of the polygenic score, depends on the phenotype, depends on the pathway of the monogenic variant. So I do think there's a lot to understand there. But people are becoming more and more open-minded to it and asking that question in their disease of interest. So I'm going to show some examples here. Hypertrophic cardiomyopathy. This is a disease of heart muscles due to sarcomere gene mutations. And polygenic score for hypertrophic cardiomyopathy stratifies survival in carriers of those mutations. And again, this is a Kaplan-Meier curve on the y-axis looking at event-free survival. And usually in HCM, this is either mortality or sudden cardiac death or having a fatal arrhythmia that's detected by a defibrillator. And the red and the orange curve are both carriers. The orange are carriers with the low polygenic score. The red are carriers with a high polygenic score. And again, the high polygenic score carriers of hypertrophic cardiomyopathy mutations had a worse event-free survival. That was published last month in Nature Genetics. Another older study, not necessarily just looking at polygenic score, but looking at the interaction of a classic monogenic disease, Hunting disease goes by trinucleotide repeats, and looking at the age of onset and asking the question, are there any common variants that actually track with age of onset? And they did found 2 loci that actually markedly either hastened or delayed the onset of Huntington disease, even among carriers. as I mentioned, our study lumped all genes together. There are other studies starting to look at gene by gene and new genes. Those are two other genes in breast cancer, PALB2 and CHEK2. And what this study did is also looked at high and low polygenic score among carriers. Here on the left side, you're seeing that PALB2 with a high polygenic score, they have a very high cumulative incidence of breast cancer, as compared to in blue, those are PALB2 carriers with low polygenic score, and the orange is all PALB2 carriers. And then you can look at the general population here, pretty low. So again, in this case, for that gene, the risk does not come down all the way to the population average. On the contrary, if you look at CHEK2, the risk and high and low polygenic score, the risk of individuals with a CHEK2 mutation who have a low polygenic score in this study was actually lower than the population average. So again, interesting to start looking at those effect sizes in monogenic and polygenic and how they play in different genes and different polygenic scores and different diseases. And the last example I'm going to show here is a study of glaucoma. This is a variant that causes glaucoma. This is a single variant and then what we're looking at here on the right, this is in non-carriers, how the polygenic score basically stratifies cumulative risk of glaucoma. And then when you look at carriers, obviously because the numbers are low, the standard error is higher. But again, there's a clear stratification even among carriers of this mutation of the cumulative risk of glaucoma. Before I end, I want to make sure I thank my mentors. When I finished my clinical part of my cardiology training, I joined Sheikh Kathawisin's lab for a year, and Sheikh had built an amazing infrastructure at the Broad that allowed us to conduct those studies. Subsequently, when Sheikh transitioned to leave the Broad, I transitioned into Ahmed Kara's lab, who's a phenomenal mentor and is driving a lot of this research in the Eleanor lab, which leads the cardiovascular disease initiative at the Broad Institute. I want to thank the NIH for my funding. I was chatting with Liang before. He asked me how long this project took, and I said that was the key project from my training grant as part of fellowship alongside other projects. But that was the main project, so it was about 2 years. And my scientific home, the Broad Institute and Mass General Hospital, and then our collaborators at the UK Biobank and then College Genomics, which allowed us to have access to the study samples to conduct this study. And again, coming back to the conclusions, integrating monogenic and polygenic risk improves genomic risk prediction in coronary artery disease. Polygenic background modifies the penetrance of monogenic disease in tier 1 genomic conditions, and polygenic background is likely to contribute to penetrance of many monogenic traits. And with that, I'm ready to take any questions.
Thank you, Akl. Very interesting talk. Do you have any question from the audience? Please unmute yourself if you have.
Hi, I have a question. I'm Sofia Stupinski, and thanks for the presentation. So one of my questions is that, have you tried to identify which components or subset of this polygenic risk score is responsible for this effect, or do you have any plans to look into that? This is one of my questions. And the other one is that when you showed some results about BRCA1 and POLB2, so I was thinking about like, there are different POLB2 or BRCA mutations have different pathogenicity. So maybe the distribution is a result of this, and there isn't such an addition of this polygenic risk scores. So what's your point on that?
So on the first part, it's a great point. That was relating to, in order to track down the mechanisms, could you start to subcategorize the polygenic score and take different components in the polygenic score and looking at the interaction? I think it's a very important question. We did some part of that. For example, what we did is we tried to remove the cholesterol effect. So we removed essentially variants around the genes of interest, and we residualized. That was one approach as a sensitivity analysis. Then another approach we did, we residualized the score to LDL levels. And in both those approaches, the results did not change. They held together. But I do think in future research, definitely there's going to be interest in kind of trying to tease down at the mechanisms. And then regarding your second question, for sure there is heterogeneity by gene, and we know that from the effect size of genes. In the current study, we lumped all genes together. Again, partly is to gain power for the numbers, but from a clinical perspective, which I think as a doctor I always kind of try to think of the clinical questions. Right now, guidelines do not distinguish between carriers of one mutation versus the other. Whether for Lynch syndrome or for breast cancer, the management is going to be the same.
Oh, okay. So sorry, I'm not a clinician. So doctors are not looking at the estimated effect of the actual mutation, or like a pathogenicity score, or the estimated effect on the structure of the protein. It's all genes in one category. I mean, not all genes, but all mutations in a gene are in one category, or is it possible to stratify the patients?
I think it's for
Must
sure.
Be possible.
Yeah, I think it's for sure. It's very important to do that work. In our study, we don't have the power because the numbers are low. I think this is what's gonna be moving in the field soon. I think a month ago there was a paper in the New England Journal of Medicine, phenomenal paper looking at all breast cancer gene mutations and looking at actually true prospective data of the effect size over time. And I think that research just needs to be done. But I agree, we in this particular study we did not do it.
Thanks. Thank you.
So it's Matt Warman. I'll ask a question. That was a wonderful talk, by the way.
Thank you. Thanks, Dr. Warman.
And the work is gorgeous. I see my colleague Christina Jacobson's on the Zoom as well. So I don't know if she's thinking the same thing as me, but there are some relatively common loss of function mutations in genes that affect skeletal properties. And I know there's large GWAS studies for bone mass and fracture risk in the population. So do you think it would be possible to set up polygenic risk scores for, say, bone density or skeletal fragility, and then apply them to patients we identify, or individuals, I should say, we identify with these more common loss of function mutations and variable penetrance fragility phenotypes? So similar to your familial hypercholesterolemia story.
I think definitely. The answer is definitely yes. And I know that in the UK Biobank, there's actually bone scans on a large number of people. So I haven't looked into this specifically, but I would imagine someone has done a polygenic score. And I think to be fair, this data I'm showing, but I've looked into few other examples. For example, I've looked into prostate cancer initial work that I was doing in a single variant that's pretty common. And I think what ends up being is the tricky thing in finding a relationship. You need to have a polygenic score that is strong enough. That's kind of the first thing. The effect size to be strong enough even in non-carriers in the general population. Because if it's a weak effect to begin with, then the effect in carriers is probably going to be negligible. We're not going to see it. Then we need enough carriers, which would be the other challenge and then within those carriers, we need to have enough people with disease. So the effect size of the carrier also needs to be a little bit, on the stronger side to have enough people with disease. So I feel if we have these three components, it's definitely worth exploring.
So who would be the person to go to? Because this is not in my wheelhouse or Christina's wheelhouse, to first ask the first question, and that is, how predictive are polygenic risk scores for either fracture risk or for bone mineral density, something that's easily measured?
Right. To be honest, I would imagine this. I have to look, but I'm happy to look after this talk, and I'll shoot you an email. I would imagine this work. Someone has done. I would imagine someone has done this. If not, it should be done, and it would be someone, someone either from our group or other groups that are doing polygenic score work at the Broad interested in this specific question. It does seem to me that it would be a straightforward work if there's an existing GWAS, then using that. If not, then running a GWAS on a trait from the UK Biobank. Within the Eleanor lab and the Kira lab, there's people doing a lot of traits. I have to say they're biased towards cardiology traits and liver traits and fat. But some examples, for example, are from like cardiac MRI. So we have cardiac MRI measurements on more than 50,000 people now. So just measuring LV, left ventricular diameter or wall thickness of the heart, and then doing generating a polygenic score, doing a GWAS on that and generating a polygenic score, and then taking that polygenic score and asking the question, does it identify people with dilated cardiomyopathy?
Right. Well, I was really amazed that there was a paper that could do that for hypertrophic cardiomyopathy, but it has to be built on those 60,000 cardiac MRIs, I'm guessing. Right?
Yeah, me too. And to be honest, this one is not actually, and that's what was surprising to me. There are other trials of echo data, but in dilated cardiomyopathy, James Percello, who's one of my co-fellows, who works with Patrick also now, has done the work and published Nature Communications. It was a dilated cardiomyopathy, and he did the same kind of study on dilated cardiomyopathy, where basically generated the polygenic score and then looked at the effect of the polygenic score within DC Titan mutation carriers.